Good morning, my name is Ashley Turner. I would like to introduce Gabby Davis. Gabby Davis is a biology graduate student in the Department of Biology. She also serves as a graduate teaching assistant, and her number one passion is veterinary medicine. So you're going to learn a little bit about that today. And her project, this is basically the basis of her thesis, and she has plans to actually graduate this summer. So without further ado, Gabby Davis.
Thank you, Dr. Turner. Okay, so the title of this is long, just bear with me. It's called An Evolutionary Comparative Study of Congenital Stationary Night Blindness Associated TRPM1 Genetic Variants of Uncertain Significance in Humans and Horses Utilizing C. elegans. Okay? So, the basis of this is genetics, obviously. So, first we need to know what a gene is. A gene is a segment of DNA, and the DNA itself is information for a certain trait, and that is transcribed and translated into proteins, and those proteins are used to show phenotypes or physical things within the body itself. So this, for example, could be used for eye color. In mine that you're going to see is going to be in horses, and it's going to be their fur coat. So depending on what it is, the gene itself is used to create the proteins, and the proteins are the ones that actually create the color, or in my case would be the fur coat of the horses itself. Okay, in this we're also looking at genetic mutations. So the genetic mutation that I'm looking at is called a point mutation or a missense mutation, and if you look at the highlighted area right here, it can actually change, and it can change the actual function and the phenotype that you see for the animal or for a human itself. So we'll be looking at a mutation that occurs in a specific gene. So my gene that we're going to be looking at is called TRPM1, like you saw in the title, and this gene is actually associated in humans, horses, and our model, which is nematode or C. elegans. In horses, the gene, whenever it has a mutation in it, or the point mutation that you just learned about, it actually is associated with a disease called congenital stationary night blindness. This is an ophthalmologic disorder in horses that also is associated with LCP, or a leopard complex, that can be seen as kind of like a spotting in some horses. So, and then for this gene itself, or this treatment, there aren't any treatments or cure for it. Okay, so because we're looking at this gene in different species, we're also doing a phylogenetic analysis to actually determine the relationship between all three of these species and the gene itself. For this, you can see the arrows. Human is at the top, and then horse is kind of in the middle, and then our model, nematode, is going to be at the very bottom. Okay. So, in the nematode ortholog that we're using in the lab to look at this disease in horses, it actually has a different name. The name is gone too, because this is an ortholog of the TRPM1 gene. So the ortholog in the nematode actually has a different function than what it's going to have in horses, and it actually has to do with vulva development and gonad cell division. So it actually affects the development for them to reproduce. Okay? Okay. So in all three of these, there are different sites for them because there are different animals associated with it, but you're all looking at the same gene within that specific animal. So what we did was we actually went in and we found the location of where this variant of uncertain significance is going to be within the gene for that point mutation to occur. And right here you can actually see what amino acid is changing whenever we look at it, and the different areas within the gene sequence that is going to be occurring for each specific species. Okay, so for this hypothesis, it's very long again, so sorry, but it says, first, if the TRPM1, this area of the gene, variant of uncertain significance is associated with congenital stationary night blindness in humans, then the genetic locus will be evolutionary conserved between humans and horses. Okay. Second, if the TRPM1 variant of uncertain significance is conserved across humans, horses, and nematodes, then the homozygous mutant in nematode will be present with a vulvaless sterile phenotype compared to the wild type nematode. So the beginning findings of this, whenever we started looking at it in the lab, we first did a gene mutational analysis to actually decide what point mutation we were going to look at within the lab itself. Sara, the person that did this before me, she actually did 31 or looked at 31 different variants of uncertain significance within the gene themselves. And then she identified 3 of them, and then from those 3 we chose the one that you saw in the slide before. Second, we did a polyphen 2 analysis. The HuMDiff score that you see right here, it actually has a range from 0 to 1. So it's a website that you go on and you plug in your information about your point mutation, and then it gives you an analysis of what the effectiveness of it or what's the significance of it, whether it could be pathogenic within your species or not. So from 0 to 1, 0 being that it's not going to be anything, it's going to be benign, and 1 being pathogenic, and the score we got was 0.775, so meaning that it would be likely pathogenic within these species. The third thing that we did was protein modeling. So we actually took the genetic sequences and we used Swiss model to create proteins for this stuff, and we came out with different structural differences and what actually occurs within the protein model itself. So within it you can actually see the structural differences. It doesn't look very small, but it's like a little stick. Okay. The next thing that we did was a multiple sequence alignment. This actually was used a website called Benchling, and this shows the difference between all 3 of them, human, horse, and nematode, and their genetic sequences themselves. So you can actually see the areas and the location where the ATA is actually amino acid sequence that creates a protein, and it's actually going to change from ATA to GTA. The change isn't seen in here, but this is just showing that the same thing is seen within all 3 species, so it gives confirmation that the same area is being affected in all 3. Okay, the recent experiments that we did with this was a DNA primer design. The primer designs are going to be used because we did a PCR analysis with it, and the primers are going to be used whenever we get into microinjection and actually change the genetic or the DNA of the nematode themselves. So the primers were used, and we actually did PCR with them and made an annealing temperature, and then we did a gradient PCR to actually determine the annealing temperature we needed to get the specific area that we needed or the specific temperature that we needed for our primers to be the most accurate that they can possibly be. Okay, this is our DNA primer design. As you can see, this is the product size right here. So we have a forward and reverse primer. So this is basically just sections of the DNA that we're choosing to actually find our specific mutation or mutation area within the DNA itself. So this is kind of like a location finder, if you would say. So we have a forward and reverse that we're going to be using. And we took this and we created a initial PCR, like I said. So the PCR itself uses a different array, or it's used in a thermocycler, and it uses different temperatures to determine the annealing process or what it's going to actually do. And it gives you these banding patterns when it comes out. So the banding patterns, if you have multiple of them, then the annealing pattern or the annealing temperature that you need isn't as accurate as what it's supposed to be. So as you can see, we did have 2 primer sets going into it in the first PCR that we did. So 1 through 3 is primer set 1, and 4 through 6 is primer set 2. From this, there are more banding patterns coming from primer set 2, so we decided not to use that primer set going forward. We stuck with primer set 1 because it was more accurate. If you can see the 3 darker bands right there, that's along the same base pair size that we were actually looking for. So it was the more accurate one of the 2. Okay. Oh, sorry. So after we decided that, we did the gradient PCR to determine the specific annealing temperature that we needed going forward, which is 1 through 8. Each lane that we used is a different temperature, and as you can see, number 3 was probably the most accurate for us, and it helped determine what temperature we were gonna use in our later PCRs once we finished our other projects. Okay, so in conclusion and future directions of this project, multiple experiments that we did supported that this gene in this specific area within humans, horses, and nematodes is usually, or horses and humans especially, was associated with congenital stationary night blindness and is likely pathogenic and warrants further investigations in vivo. Okay, a PCR-based assay has been designed and optimized for downstream use in upcoming CRISPR-Cas9 experiments to generate these C. elegans models. So we'll actually be going in and micro-injecting the nematodes that we have in the lab to see if they have a phenotypic change within them. And the phenotypic change that we'll see is either they will die off or they won't be able to reproduce. And that's what I just said. So the microinjection will actually generate in the lab, and then assessing the impact of the GON2 function by analyzing the phenotype and when it will change. Are there any questions? Yeah.
You might have said this and I missed it, but have you worked with CRISPR before?
No, I haven't. This is actually the first time that I'll be doing it in a lab.
Okay, are you excited?
Yes, very.
Strange question, but
No,
in
go ahead.
your experiment with your annealing temperatures,
Mm-hmm.
just is the PCR machine, is it able to do different wells with different temperatures, or do you have to run that like 8 different times with 8 different temperatures?
I think it does. No, it's different wells with different temperatures. So you can actually go in and change the different temperatures depending on what you want to do.
Very cool.
We have that system in the lab now, just so you know.
I just was curious, it seemed like a lot of experiments to have to do all 8 of them,
Yeah.
it takes time.
Yeah, it was all at once, thank goodness.
So this will probably show a extreme lack of
No, completely
knowledge
understand.
in biology from this standpoint. Give me an avenue, don't sell your life. So is the hope that you could manipulate this for night blindness at some point?
Eventually, not me myself, but
Oh, not you. Should be good.
well, I gotta get through that school and other stuff first. But yeah, that's the hope. If you use a smaller model within the actual lab and you're able to show the potential of what it could do within other species that are larger, like horses or humans, then it could potentially lead to further investigation within those. Yes, ma'am.
Excellent.
Any other questions?
I will quietly thank our speaker.